There are three main ways to measure protein concentration. Spectrophotometry measures the amount of photons, or the intensity of light absorbed after it passes through sample solution. Many protein-containing solutions have the highest absorption at 280 nanometer in the spectrophotometer, due to the presence of aromatic amino acids. Tryptophan has the highest absorption at 280 nanometers, followed by tyrosine and phenylalanine. The Bradford protein assay is based on the absorption shift of the dye Kumasi Brilliant Blue when it binds to protein. The sulfide group on Kumasi Blue attract basic amino acid, including lysine, arginine, and histidine, while the arines in the Kumasi Blue attract aromatic amino acids, including phenylalanine, tryptophan, and tyrosine. These interactions stabilize the dye, changing it from brown to blue. The protein concentration can be calculated by the Beer-Lambert's law. A equals epsilon times L times C. A stands for absorbance, also known as optical density, which is the ratio of incident to transmitted light through a material. Epsilon stands for molar extinction coefficient, which is an intrinsic property of the dye. L stands for optical path length, and C stands for concentration in molar. From this relationship, we can see that protein concentration is directly proportional to the absorbance. Another way to measure protein concentration is ELISA, which stands for Enzyme-Linked Immunosorbent Assay, which is used to rapidly screen for and quantify an antigen, which is the protein in a sample. First, the protein antigen is absorbed onto well in ELISA plate and coating buffer. Another buffer containing non-specific proteins is used to block free sites in the wells. The surface is then treated with solution containing primary antibody, which binds protein of interest. The unbound antibody is washed away. The surface is then treated with a secondary antibody linked to an enzyme that catalyzes a reaction that forms a color product. Unbound secondary antibody is washed away. Substrate of the antibody-linked enzyme is added, and the color intensity of the product is proportional to the concentration of protein of interest in the sample. The third way to measure protein concentration is immunoblot assay, also known as Western blot. First, SDS-PAGE separates proteins based on size. More details about SDS-PAGE has been covered in my first video about protein analysis. Proteins are then transferred electrophoretically to a natural cellulose membrane. Similar to ELISA, non-specific proteins are added to block unoccupied sites. The membrane is then treated by primary and secondary antibody, and the concentration of protein is detected by either colorimetry, radioactivity, chemiluminescence, or fluorescence. There are three main ways to measure protein-protein interactions. Co-immunoprecipitation is the precipitation of a tag protein by using antibody that binds specifically to the target protein along with other proteins associated with it. Identifying the associated proteins reveals some intracellular protein-protein interactions of the tag protein. Fluorescence resonant energy transfer, abbreviated as FRET, detects energy transfer between two light-sensitive molecules known as chromophores. The efficiency of energy transfer is inversely proportional to distance between the two chromophores. Two proteins can be labeled with fluorescence tags to track interactions between proteins. The yeast hybrid system aims to bring together the DNA binding domain and the activation domain of yeast Gal4 protein through interaction of two proteins to which each domain is fused. The DNA binding domain binds to the promoter, also known as upstream activation sequence, or UAS, and the activating domain binds to a reporter gene. Transcription of the Gal4 protein will only occur when both the bait binding to the Gal4 binding domain and the prey binding to the Gal4 activation domain are present and interacting. The yeast-1 hybrid system measures DNA-protein interaction, the yeast-2 hybrid system measures protein-protein interactions, and the yeast-3 hybrid system measures RNA-protein interactions. Besides the yeast-1 hybrid system, there are four other ways to measure protein-DNA interactions. The filter binding assay measures the affinity between protein and DNA using a natural cellulose filter, which is negatively charged. Since most proteins have a net positive charge, they will stick to the filter, and DNAs are negatively charged, so they won't stick to the filter. Only DNA that is bound by protein will stick. The electrophoretic mobility shift assay, abbreviated as EMSA, uses electrophoresis of either polyacrylamide or agarose gel to separate a mixture of DNA and proteins. The speed is determined by their size and charge. The lane with a protein-DNA complex will move more slowly. Chromatin immunoprecipitation is a type of immunoprecipitation used to determine whether specific proteins are associated with specific genomic regions. First, DNA and associated proteins on chromatin are cross-linked. Next, DNA-protein complexes are sheared into DNA fragments by cell negation or nuclease digestion. Cross-linked DNA fragments associated with proteins of interest are selectively immunoprecipitated from the cell debris using appropriate protein-specific antibody. Associated DNA fragments are purified and their sequence is determined. Finally, the DNase footprinting assay locates the protein binding site on a particular DNA molecule by using DNase, which is an abbreviation for deoxyribonuclease, to cleave the DNA, followed by gel electrophoresis to detect the resulting cleavage pattern.